In this video, we will discuss how this C1GalT1 knockdown restores sensitivity to gemcitabine in pancreatic cancer cells. In the previous video, we have understood that with C1GalT1 knockdown will cause reduce in the cell viability of the pancreatic cancer cells. So they wanted to check whether this reduced cell viability will also cause increase in the sensitivity of the gemcitabine, as the patients who are treated with gemcitabine will develop gemcitabine resistance for further treatment. Gemcitabine is a drug used for the treatment of cancer cells, used for the treatment of cancers. So first they took the cells and did the transient knockdown using guide RNAs against C1GalT1. It is silence Si C1GalT1, and they have used it at different concentrations, and we can see that C1GalT1 signals has not been shown in the panel A. They also did the stable knockdown, and those results are shown in the supplemental figures. And this knockdown they did it, and these studies they did it for HPAF2 and HPAC cells. For both the cells, we can see that transient knockdown seems to be successful, as there is no western blot signals for the C1GalT1. Thereafter, they checked the early apoptotic signals for the control Si RNAs and for the silencing RNAs against C1GalT1, and they can see that the signals for the C1GalT1 is high compared to the control Si RNAs. For this HPAF2, you can see in the first top left figure of this panel B, it increased from 4.3% to 8.8%, and when they use the gemcitabine, the increase is further, and this increase is further. This difference between this early apoptotic signals further increased. That is, when the gemcitabine treatment is done with 50 micromolar, in the control, early apoptotic signals increased to 5%, whereas in the C1GalT1 knockdown it increased to 14.2%. And similar kind of results they have seen for HPAC cell lines. With the C1GalT1 knockdown, the early apoptotic cells were 2.5%, but with gemcitabine treatment it increased to 27.1%, whereas in the control it increased to 16.4%. This indicates that gemcitabine sensitivity increased in the cells which are treated with in which the C1GalT1 has been knocked down. And these results are shown here are for the transient C1GalT1 knockdown, but the stable C1GalT1 knockdown results are also shown in the supplemental figures. And here the quantification shows in the panel C, where we can see that in the HPAF2 with the gemcitabine treatment, the increase in the early apoptotic signals is statistically significant compared to the case where gemcitabine treatment is not done in the C1GalT1 knockdown. Whereas in the case of control, the early apoptotic signals changes are not significant. And for the HPAC, the increase in the early apoptotic signals is significant, but the increase is high in the case where C1GalT1 has been knocked down. And this also give indications that anti-apoptotic factors might be decreased, as the apoptosis of this cells seems to be gives a high signals with gemcitabine treatment. So they check the anti-apoptotic factors such as BCL-XL, and they can see that these BCL-XL's signal goes down in the case where C1GalT1 has been knocked down for both HPAF2 and HPAC signals. So this further substantiates their finding that the apoptosis got increased in the case where C1GalT1 has been knocked down and the cells are treated with gemcitabine. So this C1GalT1 suppression or knockdown could be a good therapeutic target in order to increase the sensitivity of the pancreatic cancer cells against the gemcitabine. So from this results, This is one of the conclusions that seems to be plausible. So I hope you understood the results that are presented in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.